now like to move into the second part of the demo where I discuss protein structure in depth. So this lesson's learning goals is to 1, understand why protein shape matters. And it matters because it defines the functions of these proteins. Number 2 is we want to define the 4 levels of protein structure and the interactions that drive them. And finally, given a specific interaction, we want to classify that interaction within the appropriate protein structure level. So getting at that first learning goal, why does protein structure matter? And I challenge you to think of any examples where changes in protein structure is affecting protein function. So take a moment. I have 2 examples ready to go for you. So the first one is a little bit more of an everyday example. So in eggs, there's the protein albumin, which when it's fully structured is that clear goopy stuff that we see. But when we cook that protein and remove the higher levels of protein structure, they end up tangling up into those delicious egg whites. So that's one example of how protein structure is changing the function of that protein. The next example is very relevant to anybody who wants to be a physician. So that is a single amino acid substitution of glutamic acid to valine. And this substitution gives hemoglobin the sickle cell phenotype. So it's significant to human physiology. So I offer you these 2 examples so you can understand that we care about protein structure because it has such big implications on protein function. All right. So the next thing we're going to need before we cover protein structure is understanding amino acid structure. And that is because amino acids are the building blocks of proteins. Here I have a template for an amino acid structure. Take a moment to see if you can fill that structure in yourself. A little self-quiz. And here you can double check if you got it right. If not, no worries. We're going to go over it now. So all amino acids have a generalized structure where they have a constant amino group, central carbon, hydrogen group, and carboxyl group. The variable portion of amino acids falls within the side chain or R group. I'm going to be using these 2 terms interchangeably, but they mean the same thing. And depending on the identity of that R group, we get different amino acids. So going back to that hemoglobin example that we discussed, we have a glutamic acid substitution for valine. And that change in identity causes the sickle cell phenotype. So the R groups matter a lot in amino acids and also in protein structure. Okay, so now we have everything we need for the 4 levels of protein structure. So here's a depiction of them. We have 4 levels, and we're going to start right here at the most basic level, primary structure. Okay, so this is a busy slide. Let's start up here on the left together. So let's define primary structure. Primary structure can be defined as the sequence of amino acids in a polypeptide chain. So if you imagine you had a bracelet, that would be like the order of the beads on that bracelet. So the relevant interactions here are peptide bonds between amino acids. And I show you what a peptide bond looks like here. So in a dehydration reaction where we're losing H2O, 2 individual amino acids can be bonded together with a peptide bond. In this case, they're both glycine, where the R group is a hydrogen, but you can have a variety of amino acids strung together. In that order constitutes the primary structure. The takeaway for this slide, primary structure is the order of amino acids. All right, so let's get a little more complicated. Let's move on to secondary structure. So let's define it again. Local structures formed within a polypeptide chain due to interactions between backbone molecules. So that might be a new vocab for you. So let's define that. The polypeptide backbone is all molecules of a polypeptide except for the R groups. So the relevant interactions we're considering here are hydrogen bonding between this backbone amino H and this backbone carbonyl oxygen. And these 2 molecules will have dipole-dipole interactions because the oxygen is extremely electronegative. And those hydrogen bonds will form alpha helices and beta pleated sheets. So let's take a look at what that looks like. Okay, so on a single polypeptide, we get these local structures forming because of hydrogen bonding between backbone molecules. So we get the alpha helix and we can get the beta pleated sheet. So the main takeaway for secondary structure is it has to do with backbone molecules, anything except for the R groups, and it's predominantly driven by hydrogen bonding. Okay, let's go to the next one. So we just covered backbone interactions. Tertiary structure is all about R group or side chain interactions. So the definition is the overall three-dimensional structure of a polypeptide stabilized by R group interactions. So because R groups are so various, there's numerous interactions, but I'm just going to cover the major ones here. So the first one is hydrophobic or philic interaction. So here we have two valines. They have no polar molecules in the side chain, so they don't like water. So they're going to group up and try to avoid the often aqueous surrounding of proteins. That's one example of tertiary structure. We also have non-covalent bonding, such as ionic bonds and hydrogen bonds. Finally, the one prominent covalent bond that's in tertiary structure is disulfide bonds. So this occurs between two cysteines, and they will completely share their electron pair. So that's a covalent bond. So tertiary structure, the main takeaway is interactions between R groups on a single polypeptide chain. All right, let's go on to the final one. So this is quaternary structure. Some proteins will not have quaternary structure. And this is because they are fully functional as a single polypeptide. But for proteins made out of multiple polypeptides, these multiple polypeptides, also known as subunits, have to come together. And that's what this structure is about. So the relevant interactions here are identical to tertiary, but the key is that they are between polypeptides, not within a single polypeptide. So a good example of that here is hemoglobin, where we have 4 subunits, 2 beta and 2 alpha. Another common example is DNA polymerase, which is a very complicated protein at 10 subunits. So that's quaternary structure. The takeaway is between polypeptides, not within. So because I just threw a lot of knowledge at you, here is a summary of protein structure. Because we just went over it, I'm not going to go over it again, but for a student who needed to come back, needed a refresher, this would be a great slide for them to. All right, so normally I would pause here for any questions, but because it's the demo, we can dive right into the MCAT question. So here's the question. The tertiary structure of a protein is influenced by, here are options, here are answers. So before we even go into the options and answers, let's just recap what tertiary structure is and means. So the definition of tertiary structure is the overall 3D polypeptide shape formed by R group, also known as side chain interactions, on a single polypeptide. And some kinds of prominent interactions that are included in tertiary structure are hydrophobic and philic ones, non-covalent bonds like hydrogen and ionic, and the covalent disulfide linkage. So let's box up this knowledge, pop it in the corner, and then we can tackle our question. So we can go line by each of our answers. So the first one says covalent interactions between constituent amino acids. So constituent amino acids, those are within a polypeptide and could mean the R group interactions. So this passes our first check of tertiary structure. Next, do covalent interactions play a role in tertiary structure? And the answer is yes, because disulfide linkages are an example of covalent interaction. So we'd give that option a check and move on to the next one. So this one asks hydrogen bonding with side chains of neighboring proteins. So really quickly, does this pass single polypeptide chain between R group check? And the answer here is actually no, because neighboring proteins are not a single polypeptide chain. So we immediately know this option is no-go. So we can move on to the next one. This one says ionic bonding between side chains. So does this pass the check? Single polypeptide chain between R groups? And the answer is yes, because it talks about R groups here, also known as side chains, and doesn't specify between within a polypeptide or between. So we can assume that it is within a polypeptide. And ionic bonding is an example of tertiary structure interactions. So we can give that one a check. And we'll move on to the next one. So this last one just asks, generally, does the hydrophobic effect play a role? And yes, it does. So we can give this one an easy pass. So taken all together, we can look at our options. And D is the only one that satisfies the conditions that we just elucidated. So because this is a demo, I know there aren't going to be any questions, so I'd just like to thank you all for listening.